Hello, everyone, and welcome to part one of the lecture on chapter 7, microbial genetics. We're going to be covering genetics for 2 weeks, so I'm not going to do the entire chapter this week. There are a couple of terms that you will hear. Genetics, you've probably heard 1,000 times. Genome, you may already know this term, but in case you don't, I'm going to cover it here. Whoops. I don't know what I just did. Let's try that again. Share screen and come back to here. So genetics, there we go. This is the stuff, we've been studying it since Mendel, the study of inheritance and inheritable traits that are expressed in an organism's genetic material. So in eukaryotic organisms, we're always talking about DNA. In fact, in the prokaryotes, we're also talking about DNA. When we get to talking about viruses, it'll be, there's still some viruses that use DNA, but then there's another trick that some of the viruses can do. Okay, then that term genome refers to all of the genetic material in the organism. In eukaryotes, we're talking about the stuff that's in the nucleus, but then there's also some genetic information outside of the nucleus. So including all the genes. Remember, the genes are the instructions for making proteins. And the exact sequence of these A's, T's, G's, and C's, that's considered part of the genome. You'll sometimes hear people refer to genomics. Sometimes it's used as a more modern use or more modern term that includes nucleotide sequences. There's a faculty member in the nursing program that refers to genomics. I use genetics and genomics kind of interchangeably. But the genetics is the more historical term. Genomics, now that we have the technology to figure out what the A's, T's, G's, and C's, what the order of those is, and that's how we do gene sequencing. We can do things like paternity tests. We can check people for a particular genetic disorder, that kind of thing. Okay, let me remind you the structure of nucleic acids. We covered this in the chapter on chemistry. Nucleic acids are polymers. They're repeating subunits or the nucleotides. Each of the nucleotide has 3 parts to it. There's a phosphate group, a sugar, and a nitrogenous base. The difference between DNA and RNA, I'm going to remind you, has to do with the sugar. In RNA, it's a ribose sugar, and in DNA, it's the deoxyribose, which only means that on one of the carbons, instead of having an OH group, there's just an H group. When we talk about the length of the DNA, you'll hear base pairs. So remember, these A's, T's bind with each other, and the C's and G's bind with each other across the way like a little ladder. Those are the rungs of the ladder. That's what's referred to as a base pair. You know the stuff. Okay, so here are a couple of nucleotides. Well, there's the sugar and the nitrogenous base part of things. This one's adenine A. This is a double ring structure. Across the way, it binds to a single ring structure, thymine. Same deal. If we're looking at RNA, the same thing happens when we've got an A on one side, but instead of a T, thymine, we have a uracil. They're very similar to each other in structure. I'm trying to see if I can quickly identify. Okay, so the big difference between thymine and uracil is right here. There's a methyl group on the thymine and just a hydrogen on the uracil. No big deal, but they're a little bit different from each other. Okay, when we go over to G's and C's, same kind of relationship. On one side, we've got one of the nitrogenous bases is a double ring structure, and on the other side, it's a single ring structure. More details, the hydrogen bonds is what holds the base pairs together. Please note that between A's and T's, there's a couple of hydrogen bonds, and between C's and G's, there are 3 hydrogen bonds. That's why the adenine doesn't line up quite right with the cytosine on the other side. So that's why those two don't bind to each other. Okay, this is subtle, but one of the things we said was the difference between DNA and RNA is the sugar. So the sugar that's here has to be a deoxyribose. The sugar that's here has to be a ribose. This is the RNA version. And then again, what I've already alluded to is how these base pairs form across the way has to do with these hydrogen bonds. And that is the giant secret between how the DNA can get copied, and we get very few typos, and we usually make a very good copy, a very faithful copy. So this sequence is preserved from one generation to the next. The ribbons in the ribbon model is made up of the alternating phosphate and sugar, and then the rungs across the way, these are made of the nitrogenous bases. How things, you've probably studied eukaryotic organisms before. We're going to talk a little bit about them, but let's have a look at the prokaryotic genome first. The main portion of the DNA is a ring. We'll get to that. So there are some proteins, there's some RNA involved. One big difference with prokaryotic cells, they're haploid, which means they have one copy of the genetic information, as opposed to being diploid, where you have two copies of each thing. And then in the case of prokaryotes, we're generally talking about one chromosome, where in eukaryotic organisms, there's often more than one chromosome. And I haven't said it here, but we'll say it later. The single chromosome in the prokaryotes is generally a ring. The thing is a circular molecule. In eukaryotic organisms, that's often just the chromosomes are generally bar-shaped. Remember that one of the giant differences between prokaryotes and eukaryotes is the fact that eukaryotes have an actual nucleus, a membrane-bounded organelle inside the cell. That's where nuclear information is. In prokaryotes, they don't have that, but they do have a place where the chromosome hangs out. I'm going to pause this recording for just a moment. So we were just talking about this nucleoid region. So nuclei like kernel, but the oid means it's not exactly a nucleus. It doesn't have a membrane around it the way a eukaryotic cell would have. However, wherever the chromosome is, that region is referred to as the nucleoid region. And as I mentioned, oh yeah, here it is. So the DNA of the prokaryotes tends to be around a molecule that joins back to itself. It's a double-stranded DNA molecule. So here, if this is a transmission electron micrograph, think light microscope, but with electrons. So very, very fine resolution. The region where the chromosome is, that's the region that's referred to as the nucleoid. And the chromosome, just like with eukaryotic cells, it coils up and it's real tight. Here it's all coiled up and stuck in this tiny area. If you were to take the chromosome out and uncoil it, it's incredibly long. So this would be the size of the bacterium, and then there's the chromosome all pulled out. We will get to this. So there's this main chromosome, and then there are these smaller circular structures also. They're referred to as plasmids, and different bacteria have different amounts of plasmids, but it's one of the coolest things about prokaryotes. We'll get to it. All right, so let's talk about these plasmids. They're smaller molecules of DNA. They're not as big as that giant chromosome. They copy themselves independently from the big chromosome. And many of the genes that are on them, on those plasmids, they're not actually necessary for the cell to do its cellular reactions and metabolism, for the cell to grow, for the cell even to make copies of itself. However, I don't know how to describe it other than like if you have a book and you have appendices in the back, like it's a little extra information like that. And they can help the bacteria to survive in their environment. So fertility plasmids help the thing to make more copies of those plasmids. Resistance plasmids, why they have these different names depend on the genes that are located on these little rings of extra rings of DNA. So if that plasmid is carrying a gene that provides some sort of resistance, like for example, to an antibiotic or to a metal like mercury, that plasmid would be referred to as a resistance plasmid. Some genes that are carried on plasmids code for a protein that is for fighting off a different bacterium. Those are bacteriocin plasmids. And then finally, we refer to plasmids that are carrying information that are going to help the bacterium to survive and reproduce inside a host, or allow the bacterium to elude detection by the host's immune system. We refer to those as virulence plasmids. Virulence refers to like how sick can this thing make you, and it's not the bacteria are trying to make you sick, they're just trying to stay alive, and anything that's going to keep them from being killed off by the host may contribute to making the host sicker. Let's see, so let's back to eukaryotic organisms. There are chromosomes that are in the nucleus. Remember, that's the big structure that contains the chromosomal information in most in eukaryotic cells. Typically, there's more than one chromosome per cell in most eukaryotic species. And as I mentioned before, I said they were bar-shaped. They're linear. In other words, they don't circle back onto themselves, but they're hidden away, sequestered in the nucleus. And many eukaryotic organisms have two copies of each chromosome. So say we're diploid. For example, in humans, we have 23 pairs. So two copies of each of 23 chromosomes for a total of 46 chromosomes. You might remember this from your biology classes. The double-stranded DNA is not just like allowed to be by itself. The thing is wrapped around these protein balls. We used to think those protein balls were really passive. It turns out the protein balls are quite active in coiling this thing up and allowing certain genes to be transcribed and other genes not transcribed. And yeah, so the protein balls serve as, think like the toilet, like that cardboard thing on the inside of the toilet paper. It gives a place to like wrap this thing around, but more active than that. Kind of like it would determine how much toilet paper do you get to pull out, that kind of thing. So anyway, it's coiled and super coiled and then finally all bunched up. When it's time for the cell to reproduce, it bunches up all the DNA so that it doesn't lose any parts when it's time for everything to separate. Most of the time, the chromosomes do not actually look like this. This is what they look like when we take we take the pictures when the chromosomes look like this so that we can see the chromosomes more easily. In addition to the DNA that's actually in the nucleus, there are some other chromosomes in our cells. Totally bizarre. Extranuclear, that's outside of the nucleus. Where we find the most of that extranuclear DNA is in some organelles. Mitochondria have their own circles of DNA, and chloroplasts in plants and the photosynthetic organisms have their own DNA. That's weird. This is one of the things that led to the hypothesis that mitochondria and chloroplasts were at one time free living organisms that then became contained in an endosymbiosis, a symbiotic relationship between two species. Now, the mitochondria, the chloroplast, can't survive without the rest of the eukaryotic cell, and the eukaryotic cells can't survive without the mitochondria or the chloroplast. In other words, what this means is most of the genes that we have are actually in the nuclei, but about 5% of the RNA and proteins gets coded for by these extra nuclear DNA structures. And here's a fun fact. Those plasmids, those extra little rings of DNA, not contained in a mitochondrion or a chloroplast, can be found in some other eukaryotic organisms. Some fungi, some algae, and some protozoa have their own plasmids. That's fun. All right, so here's a summary table and our last slide for this section. Remember the three domains: bacteria, archaea, and eukarya. The eukarya is the one group where you'll find some diploid organisms. Everybody else is haploid. You do find plasmids in some cells on all three domains. Now notice this is an important point because a lot of people get this one confused. Double-stranded, that's what DS stands for, double-stranded DNA in all of the domains of cellular organisms. It is only when we go outside of the cellular organisms, meaning bacteria, that we get something other than double-stranded DNA. Otherwise, everybody is using double-stranded DNA. And where you find it, if we were a eukaryotic organism, we're talking about most of it in the nucleus, but some in those little organelles that we think were one time free living bacteria, the mitochondria and the chloroplast. But then there's these extra plasmids in some species. Okay. And then finally, whether histones are present or not, everybody has histones except for the bacteria. It's still not by itself. There's still some proteins involved, but they're not exactly histones. Okay, and that wraps up part 1 of the information on chapter 7 on mitochondrial genetics. All right, see you over in part 2.